was the last leg of the opening of the EAGR labs. We ended off in the Eastern Cape at the University of Fort Hare.
I am smiling from ear to ear because DIPLOMICS has extended a wonderful relationship to our faculty, the University of Fort Hare, with fantastic equipment support, which will take our research to the next level.
Okay, the new equipment include the thermocycler, the qubit, the gel electrophoretic tank, the UV viewer, gel viewer.
The University of Fort Hare and SIAB have a long-standing collaboration, and it was great to see evidence of that. The organisers of the lab have got great plans of what they want to do, and we wish them well for the future.
The current project we're working on is on keratase production for various industries, because these proteases have garnered a lot of attention within various industries due to their ability to biodegrade different keratinous materials that as a result of a lot of pollution. So this equipment will help us a lot to have hands-on work and hands-on experience with sequencing, because previously most of our sequences were done in Stellenbosch. So this will definitely help us to have that skill at our disposal.
Most of, if not all, of the ancillary equipment needed for gene sequencing related research been provided by DIPLOMICS. And that wouldn't have been possible without the wonderful support of colleagues Shane and the rest of the colleagues in DIPLOMICS.
It always surprises me how people are not used to holding a pipette or have no experience in molecular biology. But by setting up these labs with the help of SIAB as well, it's the first step to people taking their research to the next level and adding a genomics and a molecular biology component to what they're studying and further understanding the diversity that we find both in our environment but also around the population.
We really appreciate the help from YNAB. SIAB has been very, very helpful to us because we didn't have much facilities until now. DIPLOMICS came and offered some equipment. Now you have a clear image of actually what you are doing, especially when it comes to molecular biology, rather than on your research, you're doing your theory. She brought that clear image to us that we understand the genetics, we understand the molecular biology, rather than just reading and sending samples. So all the trainings, they were very helpful and impactful.
Yes, you've got equipment now, but it's very important that you realize that we're not leaving you in a vacuum. Teamwork is always important. So you need to know that if you've got questions, if you're unsure, if you have a problem, contact us. We'll do whatever we can to assist. And the nice thing about science is because it's a small community, if either myself or Shane or Mhlekazi or one of us can't answer that question, we will know somebody else who could perhaps help you.
It has made my research much easier and much interesting. Since I've experienced some things that we don't even have, I learned a lot. I learned a lot with the help of DIPLOMICS. So thank you so much, DIPLOMICS.
It's quite cool too. So we're really excited to use a lot of these.